Hey everyone, welcome back. Today, we're diving into the fascinating and often misunderstood world of cloning. With all the buzz and controversy, let's separate fact from fiction and see what's really going on in the lab. So, what exactly is cloning? Cloning is one of those topics that really gets people talking. Recently, researchers have made headlines by successfully cloning a human embryo and extracting stem cells from it. These stem cells are a huge deal because they could potentially treat serious conditions like diabetes, Parkinson's disease, and spinal cord injuries. What's the science behind it? To clear up some confusion, let's break it down. Cloning, as we understand it today, involves a process called nuclear transfer, not just splitting embryos like in the early days of cloning experiments. Dr. Harry Griffin from the Roslin Institute, famous for creating Dolly the sheep, explains, clones are genetically identical individuals. Identical twins are essentially clones. In natural embryo development, cells divide and specialize into a baby. If those initial cells split apart after the first division, you get identical twins. In the lab, cloning has evolved from splitting embryos to nuclear transfer. This involves removing DNA from an egg and replacing it with DNA from an adult cell. The result? A new individual that's a genetic replica of the adult donor. Recent advances: cloning a human embryo. Recently, researchers at South Korea's lab and the University of Michigan managed to clone a human embryo. This isn't about creating a mini-me, but rather for research, known as therapeutic cloning. They've managed to produce stem cells that could potentially be used for medical treatments. However, this breakthrough has reignited debates about cloning. While it opens doors for medical research, it also raises questions about ethics and practicality. Challenges and feasibility. The vision for cloning is to create personalized stem cells for treating diseases. But there are major hurdles. Dr. Steven Stice notes, even if we had enough eggs and funding, the technical challenges are huge. Collecting human eggs is a complex and uncomfortable process, and the costs are enormous. In the UK alone, where many people suffer from Parkinson's disease, finding enough eggs for cloning would be nearly impossible. Plus, with existing embryos from fertility treatments, there are already viable options for research without needing to clone new ones. Cloning and reproduction. What's the reality? One of the biggest misconceptions is that cloning could replace a lost loved one. But cloning isn't about recreating a person, it's about making a genetic copy. According to Dr. Griffin, only 1% to 2% of cloned animals make it to live birth, and those numbers don't translate well to humans. Clones are not the same as the individuals they are copied from. They're unique, with their own experiences and personalities. Even if technology improves, cloning won't replace someone who's gone. Identical twins, despite sharing DNA, are distinct individuals with different lives and experiences. Conclusion. So, there you have it. Cloning is a complex field with lots of potential but also significant limitations. It's a tool for research rather than a means to replace or replicate individuals. As science advances, understanding these nuances helps us navigate the ethical and practical implications of this technology. Thanks for watching. If you enjoyed this video, don't forget to like, subscribe, and hit that notification bell for more deep dives into science and tech. See you next time.